Sri Gurubhyo Namaha. Welcome to your online learning initiative, and I welcome you all for the biology class. Last class we saw about genetics, introduction, and few terms like dominant, recessive, phenotype, genotype, and allele. Now, few more terms we are going to see in detail, one after the other. Okay, so we'll move to the topic. So, terminology is very important in genetics. If you know the terms, it is very easy to study. So basic terms we'll see now. So basic terminology, what is DNA? What are genes? So we'll see first one. What is DNA? DNA is a genetic material. For example, you take a computer is there. For computer, it need OS. So what is OS? Operating system. Likewise, we have DNA as an operating system in our body. Okay, otherwise DNA we can call it as blueprint also. Okay, just I'm giving a comparative meaning. It is nothing but a blueprint. Okay, and this DNA is divided into sections and you call it as gene. In another simple term, gene is nothing but the functional segment of a DNA. Okay, next term. Just now I told about the gene. It's a functional segment of a DNA or functional unit of inheritance. In either way we can say. The term gene was given by Johansen, but the same term gene, okay, our father of genetics, what he called it as, it has factor. Gregor John Mendel called this gene as factor. It's an important one mark question. Name the scientist who called the gene as factor. Number one mark question you can ask. Another one, who coined the term gene? It's none other than Johansen. Johansen coined the term gene, and the same gene was called as factor by Mendel. Okay. Then next, allele. We were discussing about the allele. I just wrote there in the dominant and recessive, capital T, capital T. Just guess children what it is. It is tall. And then small t, it is nothing but dwarf. We are expressing with your genotype. And this pair of letters, you call it as allele. So the alleles are nothing but forms of gene. And there are two forms of gene, one dominant, another one recessive. So here you can see dominant and recessive. So I'll write nearby also. So the dominant is nothing but capital T, and the recessive nothing but small t, small t. There is one more form also that you call it as multiple allele. Okay, it is a single. Okay, multiple means many. Many alleles will be there for one characters. Might be the size or the color or the coat color. Coat color, when I say, it is about animals, fur color. Okay, this we are going to see later. So we have seen a term gene, another one alleles. In allele, a new thing called multiple alleles. So phenotype, I've discussed this already in the previous class. Phenotype, the physical appearance. By seeing, you are saying it is a tall, it is a dwarf, that is physical appearance. Genotype, expressing their genes. Okay, it is nothing but genetic makeup. You can see here physical appearance, and genotype is nothing but genetic makeup. So what are chromosomes? The chromosomes are, okay, a rod shaped structure like this. We saw this one while unfolding the gene, right? A rod shaped structure, okay, which are in a pair, okay, that carry the gene and encoded by a DNA. I'll show you the chromosome now. Just see. This is the structure of chromosome. Okay, and you can see something compressed and coiled structure. It is nothing but DNA. Okay, if you want, I'll write it. It's DNA. And this you call it as arms of chromosomes. Next, what is DNA? DNA is the deoxyribonucleic acid. It is a nucleic acid. In 11th standard and 10th standard, we read about the codes which is present in this. It is nothing but 4. Okay, that 4 you call it as adenine, I'll write here. Use a different color. Okay. We read about this term A, T, C, G. Okay. The same term, detailed manner we are going to see in next lesson. Okay. As I'm introducing the terms, I'm touching the other area also where all it involved. The DNA is a double helical structure. It composed of, see, it is a double helical structure. It is composed of codons like this. And the codons are nothing but A, adenine, T, thymine, C, cytosine, G, guanine. In a simple term, A, T, C, G, you call it as, right? It is composed of nucleic acid. Those molecules encode the genes. Encode, we are, that codons I'm talking about here. A, T, C, G. Allow genetic information to be passed to the offspring. Based on this only, there is a protein makeup, and based on that only, inheritance takes place. Next, a very interesting term, mutation. I told about the COVID-19, okay, how it get transformed from a harmless to harmful. It's all based on this only, mutation. In a simple term, mutation is nothing but sudden change in the gene. Okay, in a very simple term, I'm saying. I'm not saying a scientific term also. In a very simple term, it is a sudden change in the gene. So we'll see the scientific expression, okay, or explanation. A permanent inheritable change. See the letter, see the word permanent. It's okay. The change, what happened? It is permanent. Inheritable. It can be transferred from one generation to another generation. So how? It can be in a single gene. Okay, the mutation can happen in one particular point. If there is a DNA, one particular point, there might be a mutation. Okay, that you call it as particular point, I'm saying, no. So it is nothing but point mutation. It occurs only in single gene. That results in the existence of two or more alleles. So it disturbed in that particular point, and because of that, it is expressing with different number of genes. That is different and many number of genes. Okay, not the same. Again, I'm using the word different and many number of gene, right? So occurring at the same locus. So what is locus? Well, I'm drawing here a chromosome. Okay, the point, particular point. Again, I'll draw in pair. I'm drawing here a chromosome. Again, a particular point, the point mutation takes place, I'm saying, no? Here there are gene, and that you call it as locus. Locus is a place where the gene is placed. Okay. Now, example they are given. Because of that only, there is a polymorphism. Poly means many, morphism means body design. There are many body designs and there are many characters also. And here we have taken an example for blood group. Okay, blood group we have A, B, AB, O, like that, right? So we'll move to the next term. Homozygous, heterozygous, like phenotype, genotype, how it is coming in a pair. Another one, dominant recessive, how it is coming in the pair, and they have a difference also. The same way you learn this in a pair with a difference. Homozygous, heterozygous. Homo is nothing but same. Hetero, different. Okay, so we are going to see what is that homo and hetero. The situation in which allelic gene are identical. Okay, again I'll give you an example. So by this time you are very much familiar with this capital T and capital T. It is nothing but tallness. And that capital T, nothing but what? It is a genotype, right? And it is occurring in a pair. And you can see the same letter. Both are in uppercase. So it is homozygous. And the same thing I'm going to write like this. So it is a genotype. Whenever I'm writing with a letter, it is a genotype. It is nothing but tall. The genotype for tallness only it is, right? But here you can see two different letter. Yes, one is small letter, another one capital letter. When two different alleles are there, that you call it as what? Heterozygous. Hetero, different. Homo, same. The situation in which allelic gene are different. Here they are identical, right? Locus. Previous slide I told about the locus, the particular point. Okay, the same thing here. The location of a allelic gene. Sorry. the location of the allelic gene on the chromosome is called locus. Again, once again, I'll draw only one norm of the chromosome. Here is a gene located, means this you call it as locus, right? Now the next, I'm going to introduce one after the other the different types of cross. Okay, so far we read the terminologies, and the before class I just introduced what is genetics and about Mendel, some contrasting characters, their expressions. Now we are going to see the next stage of the lesson, various crosses. We will start with the basic cross like back cross and test cross. This you have not studied in 10th standard, so listen carefully children. Back cross and test cross. First, whenever we are learning something, we should describe the term. From the term, we have to pull out the answer, right? So, what is back cross? The name itself says back, get back something. You are going to get back the character of the parental characters, right? Test cross. Again, the name itself says what? Test. You are going to test and find out what type of genotype it is. Okay. So, we will see one after the other. Backcross is a cross of F1 progeny back to one of their parents. Okay. So, F1 is nothing but the offspring. Okay. This offspring they will take it. Okay. And they are going to intercross with any one of the parental characters. Okay. Any one of the parental character. It may be the male or female. Most preferably, they are going to select the dominant character. They want the dominant character. The outcome should be the dominant character. So for back cross, they will use a dominant character. Okay. And test cross, they are going to use a recessive character. Okay, keep in mind, back cross, you're going to get back the characteristics of the parent, and mostly they will prefer for the dominant thing. So they are going to take the parental characters and they are going to give back. Right. Next, what is test cross? A special back cross to the recessive parent is known as test cross. Here you are going to intercross with the dominant thing, whereas in test cross you are going to intercross with the recessive thing. That alone you keep in mind. That is be very easy to pull out the answer, right? This method was devised by Mendel. One mark question: who devised this method? Back cross and test cross. Why? To determine whether the dominant phenotype is homozygous or heterozygous. Okay, for that only he used this back cross and test cross, right? So what is the main difference between the test cross and back cross? It is a 2-mark question, and the cross they may ask in 1-mark question also. How will you find the genotype? Okay, like 1-mark question. The main difference between test cross and the back cross is that the test cross is used to differentiate the genotype of an individual, okay, which is phenotypically dominant, whereas a back cross, this is, they are talking about the test cross, and here they are talking about the back cross. A back cross is used to recover an allied genotype, an allied variety they are going to get, most preferably dominant, I said, from a parent which bears an allied genotype, right? So I will explain you with an example. Just check, and I want everyone to make a note of it. So back cross we will divide like this. Okay, here back cross. Okay, in back cross what we are going to do, we are going to take the F1 generation. Okay, it may be the same. I'm going to use the plant. As you are familiar with the size of the plant, tallness capital T, dwarfness small t. So I'm going to use a heterozygous thing, and this I'm going to intercross with a dominant. This is my preferable character, and This only I'm going to intercross with whatever the F1 offspring it will be, right? I can intercross with the small t also, with capital T, capital T. Okay. Again, you keep in mind, in a backcross, I have to intercross with a dominant character. I'll go back the previous slide and I'll show you. A backcross is a cross of F1 progeny. F1 progeny, the first generation of the parental offspring. Okay, that you are going to back to one of their parents. Okay, and we are going to always prefer for a dominant character. So here, this is the F1 offspring, and here I'm going to intercross with what? A dominant character. This is fixed. I have to intercross only with the dominant character, okay, of F1. The F1 may be like this. Either it is in a heterozygous condition, or in a homozygous recessive condition, or it will be in a homozygous dominant condition also. It can be in any three. But my intercross should be what? With capital T and capital T. It is nothing but the tallness. And it is homozygous dominant. This only I have to use. And the expected ratio what you will get is 1:1. In the next slide, I'm going to do the Punnett square. Punnett is a scientist who gave this method, like a checkerboard method. I'll just show once. See, like this checkerboard method they gave for the easy calculation. We'll complete for the test cross also. Test cross, if you take it, I said already, for test cross, we are going to take the recessive character, right? Okay, here I'm going to use the recessive character. The name says what? Test. Test whether it tests the genotype, whether it is a dominant or recessive, heterozygous or homozygous. So whatever the condition, I have to intercross with small t, small t, small t, small t. Okay. The condition may be like capital T, small t, small t, small t, capital T, capital T. Any condition I have to intercross only with small t, small t. It is nothing but the recessive character. Okay. Yes. And why we are doing this one? To find the genotype. Okay. To find the genotype, we are using this method. So we will go to the cross now. And one example I'll tell you. There is a plant which bears a purple color flower. Find out the genotype of the particular plant. Okay. How will you find the genotype of the particular plant? Either you'll go for the back cross or test cross. Think. We will go for the test cross because the question says in the beginning, find out the genotype of the plant which bears purple color flowers. Okay. So I have to find the genotype. So I should go for a test cross. Okay. How will I find? It is a purple color flower. The name itself says purple. So I have to use capital P, or I can use capital P, small p. Both what color? Purple color. I'll write here. Both purple color. Right? So this we will see in the next slide. Now first we'll go for the back cross. So in back cross, I'll change the color. Here the male and the female character will not show much difference here because it is plant. When it comes to animal, you have to be very careful in denoting the male and the female. Here I can take anything as a male and female. Okay. So here the character is capital T, small t. Okay, the F1 offspring. I'm talking about back cross here. And then with what I have to intercross? I have to intercross with the dominant character. Capital T, capital T. Now intercross this capital T, and then capital T, small t, and then here this one capital T, and then capital T, small t. Okay, so here this is tall and this one also tall. right? And here it is also tall, and this one also tall. So what we are getting? We are getting elite variety, everything tall. Again, we'll recheck once. I'll show with my cursor capital T tall, capital T small t tall, because there is a dominant character. That dominant only will express, will mask the other recessive character. Again, capital T tall. See this one, along with this you will write here. Capital T small t tall. So you are getting 100 percentage what? Tall character. Okay, you're getting the elite group. Now, the next one, if you take test cross, same, let me take it. Okay, capital T. So here I'm giving as a male character, and here I'm giving as a female character. Same, heterozygous. The size of the plant, I don't know, unknown genotype. My known genotype should be what? A recessive character. So I'm going to intercross with the recessive character. Capital T, small t, small t, small t, capital T, small t, then small t, small t. Is it right? We'll check once. Capital T, small t, small t, small t, capital T, small t, small t, small t. Yes? Check once you check here whether the letters are right. Yes? Okay, now how much you're getting? The genotype here. 2 you are getting, and here also 2. Okay, 2 what you are getting? Capital T, small t, and this too, small t, small t. I'm just writing the same thing here. Okay, the number. So this 2 is to 2 is nothing but 1 is to 1. So this is the ratio. What is the test cross ratio? 1 mark question. Name the test cross ratio. It is nothing but 1 is to 1. Okay. And this is monohybrid because we have taken only one character. Dihybrid means 4 you will get. 1 is to 1 is to 1 is to 1. Okay, that we will see later. Thank you, children.